fast forward. Oh, don't make that face. I promise this is easier than what you've been doing. At least for a minute.
No.
Yeah, because we did 8 and 9 early, right? 7, 8, and 9 we did early. We skipped from like 2 to 7, 8, and 9, and then we did 3, 4, 5, 6. Just got done with those. So back to 10, and we'll kind of go mostly in order here. Because 10 is mitosis 11 and 12. 11 and 14 really for genetics. I guess we don't go completely. And then 12 and 13 are digging into DNA. And then 15 through 17, I think, are evolution. And that's what we got to get done this year. Let's see what time, what kind of time we have left for other stuff. Anyway, tell me about mitosis. Tell me about cell growth and division. Yeah. There's some phases we're going to have to know. Yeah, you know that. Have you had to memorize them before? Yeah. Okay, so that part won't be that hard. Slightly more challenging maybe will be actually being able to recognize them under the microscope. So we'll need to do that. We'll need to actually be able to look at a microscope slide that has a bunch of cells going through mitosis, frozen, like not live, but like having been stopped in a particular phase. Look at it and go, oh yeah, that's prophase or that's metaphase or whatever. Yeah, okay. Why do they do that? Why do they divide? Okay, so give me a situation in which a cell has to divide. I like it. Yeah, you hurt yourself. There's a break in your skin, and that's absolutely true that you have these hormonal controls that suddenly kick in and go, okay, a bunch of cell division has to happen right here. Good. And it knows if what's weird is it knows to start dividing, and then it knows to stop. Like right when everything is healed over, bam, everything stops. Totally cool. You can do that in a petri dish. You can put skin cells in a petri dish. They will divide until they are exactly one layer thick, and then bam, they'll stop. Super cool. Okay, what else do you know? This is, by the way, this is what's his name? The dog, the puppy killer. Virchow's thing, right? Cells always come from other cells as part of the cell theory. So when you do it, when you make more cells, when cells come from other cells in your body, we call it mitosis. And that's what we call it in pretty much all eukaryotic cells, the complicated ones, right? The ones with a nucleus, because technically mitosis is just the division of the nucleus. It's not the division of the rest of the cell. That has another name. We don't call it mitosis if it's a prokaryote, because prokaryotes don't have nuclei. It's a simpler process in a prokaryote. It's called binary fission. It's much like what your mitochondria do. Mitosis is, we call it the M phase of a larger cycle that all cells are in, called the cell cycle. M is just one part of that. And before a cell goes into the M phase, when it's in the rest of the cell cycle, there's a couple of things it has to do with its DNA in order to proceed into the M phase. Two things specifically that a cell has to do with its DNA before it starts mitosis. What do you think? What are the two things a cell's going to need to do with its DNA before it splits? Sam. It's going to need to copy all of it, right? Whatever you got DNA-wise, whatever your genome consists of, you're going to need an extra copy. You're going to need 2 full sets of DNA so that when you divide the cell in 2, each new cell has its own full set. Perfect. It's going to need to copy its DNA. The other thing it's going to have to do, and this actually happens in the first phase of mitosis. The problem with DNA, which you now at least kind of know, is a code for what? What is DNA code for? Oh, it's coming. You're going to know what and exactly how DNA makes and codes for what it makes. DNA enlists RNA as a helper molecule and sends its code via RNA out of the nucleus to the ribosome to make proteins. Good. Yeah. Well, when it's doing that, when it's doing its job, it's all kind of yarn-like. It's in a form called chromatin, which is one of those cases of naming something before you actually know what it is. It's named because it just means colored stuff. And that's because the early stains we used on cells in early microscope days stained the DNA and not the other stuff. So we called it chromatin. Chromatin is what we call the unwound version of DNA. It's all just like, it's a big ball of yarn. And if you copy that big ball of yarn, great, you got two balls of yarn, they're all tangled up with each other. You're not going to just divide the cell and be able to get the full set on each side. You're going to mess it up. So what you got to do first, after you copy the DNA, but before you actually pinch it off, you're going to have to organize it. You're going to have to make sure it splits cleanly and you get a full set on each side. And so that's what chromosomes are all about. Chromosomes are basically the neat little packages. Chromosomes are not functional DNA. By the time you've got chromosomes, they're not doing the stuff that they do in an everyday cell. By the time you got chromosomes, you're in full-on cell division mode. Chromosomes are little X-shaped, densely packed bundles of DNA built to split. They are the way they are, X-shaped and densely packed, so that you can easily line them up, split them in two, and have exactly the full set on either side. So a chromosome, of which you have 46 in a normal healthy cell. We draw it as an X shape with kind of a dot in the middle. We're about to, you're in the beginning of a lot of words that start with C, by the way. Chromosome is one, chromatin is another. Centromere is what we call the point at the middle of a chromosome. Divide a chromosome directly down the middle through the centromere vertically, and what you have are what we call two sister chromatids. There's another C word. Cool, so that's the thing you make out of your DNA. And we're going to take that nucleus and mess with it so that we divide it, make two nuclei. Then we'll divide the cell. Is this something all your cells are doing? Are all your cells dividing all the time? Dominic, you say no? No, that'd be crazy, wouldn't it? All of your cells dividing all the time would be you as a big undifferentiated ball of slime. You'd just grow outward and just get bigger and bigger. You have very precise control. I mean, you said it with the cut thing, right? Such precise control that, you mess it up and it divides till the wound is closed, and then bam, it stops. You got perfect control over it. Are there places in your body where mitosis is constantly happening? Where would be one? Can you think of one? The lining of your stomach, absolutely, is a place where constant mitosis is happening. You're constantly replacing the stomach lining. Yes. Where else? Constant cell division. Liver. Maybe parts of the liver if it's regenerating, maybe. Which part of the body do you know? Ooh, yeah, you guys have got some cell division going on that I don't. Yeah, the ends of your long bones and your muscles, places. If you're growing, that's absolutely mitosis. That's making more cells that belong to you, so you get bigger. It's not that the cells get bigger, right? It's that you make more of them. Okay? Your heart isn't actually where more blood is made. Anybody know where blood cells are made? Bone marrow, yes. Constant cell division going on there. You've missed the biggest one because you got distracted away from it, and it's Will's fault. Yeah. Yeah, but not the surface, but like at the base of the skin, like a few layers down, right? Because you know about the outside of your skin, right? We talked about that. It's your suit of armor between you and the bad world, like it's dead, all of it. It's just a layer of dead cells to protect you. And they die on purpose, but they have to be made first before they die, right? So you've got this deeper layer of skin that's just constantly turning over new cells, constantly going through mitosis, sending them out to die on purpose to be your protection. So yes, there are places where their cells constantly going through mitosis, but there's other places where the cells get to a certain point and then they just pause. And they do the stuff that cell's supposed to do, like your brain, for example. You pretty much got all of those. You're going to take care of them because that's it. They divided when you were like a fetus and divided maybe a little more when you were a baby. And then you had all the cells you're going to have. And you're just, now when you learn stuff, it's just about making new pathways. You're not growing new cells there. So not every cell is going through mitosis all the time, but every cell is going through some phase of this called the cell cycle. So this will be the last thing we do. I would say maybe don't, if you're going to draw it early here, draw it big. Not a ton of wording, but it's good to have it big. All cells in your body are somewhere in this cycle. Yes, Sam?
I was going to ask, what happens if the brain cells are damaged?
So there is a small, it's not as true as they thought that you have all of them you're ever going to have. There is some regeneration that way, but mostly it's what I said. It's if something gets damaged, you got to build new pathways. Like if you destroyed part that's on a certain pathway, your brain has what's called plasticity and can route around stuff in the brain and build new pathways. And so most of brain healing is about that and not about making more cells. Good question. So this part only is mitosis. The rest of it, which we will name these phases separately, but together they are called interphase. So draw that correctly so that it's just these three that are called interphase. When a cell gets done divided, it enters the first phase of interphase when it's faced with the problem that, look up here, if you do this, if you're a normal sized cell and you do this, your first problem is you're half the size you're supposed to be, right? So G1 is growth, growing back to the normal size. Second phase of interphase is where you address the problem of the next division by copying all that DNA. S phase, we call it, for DNA synthesis. That's DNA. Finally, the third and final phase of interphase is mostly just prepping for mitosis. It's called G2. And we're talking about, making enough ER and Golgi and lysosomes and all that stuff, making sure there's enough parts for 2 cells worth. Last but not least, and the thing we'll spend most of this chapter on, mitosis. Anybody know the phases in order still? Almost perfect. You just switched metaphase and prophase. Prophase comes first. Pro. Anyway, prophase, metaphase, anaphase, telophase, correct. That's the division of the nucleus part. Then there's this last event called cytokinesis. That's the part where the cell actually splits. We'll save that for tomorrow. We'll save that for tomorrow because I want to give you some time in class today to finish up your Antarctic warming projects, to talk to your group members if you haven't been, to make sure and pull these flow charts together so they're as correct as you can make them. Remember, you can do it either digitally by dragging the arrows around or on paper and take a picture and upload it. But all of this stuff is due tomorrow. You did get an extra day thanks to one of your classmates making a well-reasoned argument that that's probably what I should do, so I did it.